Hi, my name is Jolie Carrillo, and I chose Dr. Jennifer Laffin for my bio role model project. She has a PhD in genetics and is currently a professor here at the U's Medical School in the Department of Laboratory Medicine and Pathology. She's currently involved in clinical genetics. I was lucky enough to set up an in-person interview with her for this project. So, Jennifer initially realized she liked science in 3rd grade when she came to the realization that she naturally excelled at it. She went through school and was most into her science classes, especially human biology. In high school, her plan was to go to medical school after college. Research wasn't even on her radar. She first went to St. Benedict/St. John's University. It wasn't until college when her chemistry professor, Henry Kucaba, talked about his research on clotting factors in class that she realized it'd be something she's into. After talking to him, she decided research was something that she wanted to do. Unfortunately, despite her interest and effort, there were no research opportunities at St. Ben's for her. She decided to transfer back home to her home state of Wisconsin and go to UW-Madison. Her original intention was to be a biochemistry major like her chemistry professor, but she ended up becoming a genetics major as it would get her out the fastest. Fun fact, she was not a big fan of genetics. She always associated it with Punnett squares and the Mendel pea plant. At the UW, she finally got jobs in labs feeding fruit flies and cleaning glassware in a chlamydia lab. While these were jobs in the lab, she wasn't fully satisfied doing grunt work. After receiving her bachelor's in genetics at the UW, she went to the University of Iowa for her PhD. Here she wrote her dissertation about identifying genes involved in heart development and congenital heart defects using express sequence tags, or ESTs, from developing rat hearts. In this paper, she compiled over 12,000 non-redundant ESTs from 30,000 sequences by analyzing cDNA libraries from embryonic and postnatal rat hearts. The libraries included microdissected tissues from key developmental stages. Over 6,000 novel ESTs were discovered and added to the NCBI rat unigene collection throughout her experimentation. After earning her PhD in genetics at the University of Iowa, she did a clinical pathology fellowship at the university. Fun fact, throughout her time in school, she played the cello semi-professionally. She was involved in the Madison Symphony, St. Cloud Symphony, and a Baroque group. Due to unfortunate circumstances, she left the fellowship at Iowa and joined a clinical cytogenetics fellowship back at the UW. After her fellowship, she worked at UW-Madison as an associate professor in the Pediatrics Genetics and Metabolism department. Then, as I mentioned in the beginning, she's now a professor here at the U's Med School as of January 2023. One of her more recent scientific papers is titled Analytical Validation of a Novel Multi-Target Blood-Based Test to Detect Hepatocellular Carcinoma, or HCC. The study it covers validates a multi-target blood-based test for detecting hepatocellular carcinoma using DNA methylation markers, alpha-fetoprotein, and patient sex. The test demonstrated high reproducibility, no significant interference, and strong analytical performance, supporting its potential for early non-invasive HCC detection in at-risk patients. A figure from the paper that presents experimental results is Figure 2, which shows the linear range for the 3 methylation markers used in the multi-target hepatocellular carcinoma blood test. The linearity study aimed to establish the range across which the methylation markers can be accurately quantified. The researchers prepared a 2-fold serial dilution series of synthetic DNA carrying the methylation markers, spanning from 5,100 copies to 10 copies per reaction. They used quantitative PCR with long-probe quantitative amplified signal chemistry to measure methylation levels. The data was analyzed by fitting different polynomial regression models, determining whether a linear model best described the relationship between DNA input and signal output. As seen on the slides, you can tell what the axes are labeled and what the trend shows. The main conclusion of the figure was that the data confirmed that the methylation marker detection was linear over the tested range, meaning that the test provides accurate and proportional quantification of DNA methylation levels within this range. This result supports the assay's robust performance and reliability for detecting low-abundance cancer biomarkers in blood samples. This slide shows a synopsis of the current research Dr. Laffin is working on. Unfortunately, I don't have enough time to go over it all, but please pause and read. It's pretty interesting. On this slide, you can see a list of mentors/teachers that were important to Dr. Laffin throughout her life. Overall, Dr. Jennifer Laffin is a scientist I admire because of her realistic journey. She started out kind of lost, which is how I felt when first approaching college, and then she found her passion. Even though she originally wanted to go to medical school and didn't enjoy genetics at first, she remained open to new paths. She embraced genetics and went on to make significant contributions in the field. Also, she not only conducts groundbreaking research but teaches and mentors future scientists, inspiring others to pursue careers in genetics and clinical medicine. Thank you for watching.